Good afternoon. My name is April Kriebel, and I'm a graduate student in the lab of Dr. Joshua Welch. Today, I'll be presenting on non-negative matrix factorization integrate single-cell multi-omic datasets with partially overlapping features. Current dataset integration methods function by subsetting the datasets that they're integrating to features that are shared between datasets. This is a methodology used by Liger, Surratt, and Harmony. And you can see here that we have to subset to the number of genes that are shared between these datasets to perform the integration. However, this entails the discarding of pertinent information, such as intergenic peak information when we're dealing with ATAC sequencing data, as well as unshared genes from single-cell RNA-seq when integrating with spatial transcriptomic datasets that are typically characterized by few genes, as well as in areas of cross-species analysis where we have non-homologous genes. Our solution to this problem is to append a U matrix into the Liger algorithm such that unshared features are incorporated in each iteration of the matrix decomposition. The unshared feature space assumes nothing about the relationship between shared and unshared features and can consequently incorporate additional genes, intergenic features, and other features that might be of interest. The matrix decomposition functions such that the HI matrices are the cell factor loadings, the VI matrices are the dataset-specific metagenes, the W matrices are the shared metagenes between the datasets, and the UI represent the unshared metagenes. To prove the efficacy of our method, we used UINMF to integrate a spatial transcriptomic dataset that was characterized by few genes with a high-quality single-cell RNA sequencing dataset. You can see in the top right the results of the original Liger algorithm that was unable to incorporate any genes that were unshared between the two data sets. And you can see the result of our proposed method here in the bottom right, labeled UINMF. We were able to use the UINMF algorithm to incorporate an additional 2,775 genes that were present in the single-cell RNA sequencing dataset, but not present in the spatial transcriptomic dataset. We also introduced a vectorized lambda parameter of 10 for the spatial transcriptomic data and 1 for the single-cell RNA sequencing data. To quantitatively prove that our method was beneficial to the integration, we compared UINMF to existing algorithms using ARI and purity scores. We used the drop phase or the single-cell RNA sequencing data as ground truth because this was high enough quality that it could typically be used for cellular annotation. And you can see that in the results, our method is statistically significant over both Surratt and our existing INMF method. To explore other analysis spaces where UINMF might be beneficial, we performed a cross-species integration analysis between a single-cell lizard dataset and a single-cell mouse dataset. As our shared features, we used the 1,979 homologous genes between the two species, and for our unshared features, we used 166 non-homologous lizard genes. You can see the results of our integration in the UMaps on the right. The top UMap illustrates the alignment that we saw between the two datasets. In blue, you'll see the mouse cells, and in pink, you'll see the lizard cells. The bottom two UMaps show the different species annotated by their original cell types. So, for instance, this is the lizard cells annotated by their original cell types, and these are the mouse cells annotated by their original cell types. And you can see appropriate cell type correspondence between many of the cell types, such as the astrocytes, the inhibitory neurons, as well as the excitatory neurons. We further wish to quantify the benefit of using UINMF over INMF, which, again, cannot incorporate the unshared genes. And you will see that we did this using both the mouse and the lizard cell labels. You can see that we saw a significant improvement in the ARI scores using both the mouse and the lizard labels, and we saw significant improvement in the purity using the mouse dataset. We did not, however, see significant improvement in the lizard purity, but you can see that we had a general trend of improved scores. Overall, we believe that we've demonstrated that UINMF can improve dataset integration by using unshared features. We think that other potential uses of UINMF apply to the intergenic peaks when integrating single-cell ATAC and single-cell RNA sequencing data, more cross-species analysis and spatial transcriptomic datasets, as well as integration of dual omics datasets with spatial transcriptomic data. And we look for future extensions of UINMF with integrations of high-C data as well. Thank you, and I'm happy to take any questions that you may have.